Hello everyone. Chapter 6, Expression of Biological Information. 6.3 Learning Outcomes. At the end of this video, students should be able to explain briefly transcription and translation. And also, students should be able to introduce codon and its relationship with sequence of amino acid using genetic code table. Okay, let's start. Generally, DNA undergo transcription to produce RNA in the nucleus, then this RNA goes to the cytoplasm to be translated into protein. The process of making protein starts with transcription. This process occurs in the nucleus and catalyzed by RNA polymerase. In this process, the mRNA is synthesized using single DNA strand as template. Only a part of DNA will be copied. There are 3 steps in transcription: initiation, elongation and termination. During initiation, RNA polymerase bind to the precise location and orientation on the promoter. This in turn determines where transcription starts and which of the 2 strands of DNA helix is used as the template. Then, RNA polymerase unwinds the 2 DNA strands and begins transcribing the DNA template strand at the start point. In the second step, which is elongation, as RNA polymerase moves along DNA, it untwists the double helix, exposing about 10 to 20 DNA nucleotides at a time for pairing with RNA nucleotides. These enzyme also adds free RNA nucleotides to the 3' end of the growing RNA molecule as it continues along the double helix. A single gene can be transcribed simultaneously by several molecules of RNA polymerase. The congregation of many polymerase molecules simultaneously transcribing a single gene increases the amount of mRNA transcribed from it, which helps the cell make the encoded protein in large amounts. The last step in transcription is termination. Bacteria and eukaryotes differ in the way they terminate transcription. In bacteria, transcription proceeds through a terminator sequence in the DNA. The transcribed terminator functions as the terminator signal, causing the polymerase to detach from the DNA and release the mRNA. In eukaryotes, RNA polymerase transcribed a sequence on the DNA called polyadenylation signal sequence, which specifies a signal to cut the RNA transcript free from RNA polymerase, releasing the pre-mRNA. The pre-mRNA then undergoes splicing process. How is pre-mRNA splicing carried out? During this process, certain interior sections of RNA molecule or non-coding region called intron are cut out, and the remaining parts or coding region called exon, spliced together producing an mRNA molecule that ready for translation. The removal of introns is accomplished by a large complex made of protein and small RNA called spliceosome. After further modification, the mature mRNA travels to the cytoplasm, where it directs protein synthesis. Let us review together the function of RNA polymerase. This enzyme catalyzes the selection on which gene to be transcribed. Recognizes one of the 2 DNA strands it should copy or act as template. It also identifies where it should begin and end the transcription. The enzyme also catalyzes the unwinding of DNA double helix by breaking the hydrogen bonds. RNA polymerase catalyzes the addition of new RNA nucleotides from 5' to 3' direction. At the end of the process, this enzyme catalyzes the rewinding of the DNA strands. You can access the questions by scanning QR code below. Before we proceed with translation, let us find out about codon, anticodon and tRNA. Codon is a triplet of nucleotides within mRNA that codes for an amino acid. Codon are customarily written in the 5' to 3' direction. There are 6 characteristics of codon. One is each codon consists of triplet nucleotide combination. There are 64 codons in genetic code table, but only 61 codons specify amino acids. 2. There is only one start codon, which is AUG, that encodes for methionine, MET. 3. There are 3 stop codons, which is UAA, UGA, and UAG. This codon indicates the termination codons and do not specify any amino acids. 4. The reading of codons are non-overlapping. As example of sequence, A, U, G, U, C, U, A, G, U, read as AUG, UCU, AGU. Not as AUG, GUC, CUA. 5. One codon is specified for only one amino acid, but one amino acid can be encoded by more than one codon. Anticodon is a triplet nucleotide located at 3' end of a tRNA molecule. This anticodon pairs with a particular complementary codon on an mRNA molecule. Anticodons are conventionally written 3' to 5' to align properly with codons written 5' to 3'. Transfer RNA or tRNA consists of single RNA strand, sized, about 80 nucleotides long. It has cloverleaf structure, where anticodon, on cloverleaf end, while amino acid, attached on 3' end. The function of tRNA is to transfer the amino acids, encoded by codon, to the ribosome. So, what is the relationship between the codon and anticodon? During translation, codons will pair with anticodons, so that the correct amino acids can be linked to a given codon. I shall repeat. Codons will pair with anticodons, so the correct amino acids can be linked to a given codon. These are how the codons match to the amino acids. For this example, AUG in the mRNA is the codon. It will pair up with UAC, which is anticodon that carried on tRNA that linked to amino acid, which is MET. However, this codon, amino acid matched, will be provided in the genetic code table. So, don't bother to memorize them. These are the genetic code table. How to read these table? You will learn it later in the tutorial class. Now, we are going to learn further about the translation. Translation is a process of synthesized a polypeptide using the genetic information encoded in an mRNA molecule. The process occurs in the cytoplasm and facilitates by tRNA in ribosome. But before translation can take place, each amino acid has to be matched with the correct tRNA by a specific enzyme called amino acyl tRNA synthetase. This process is known as activation of amino acids. There are 3 steps in translation: initiation, elongation, and termination. During initiation, the start codon AUG signals the start of translation. This is important because it establishes the codon reading frame for the mRNA. In eukaryotes, the small subunit, with the initiator tRNA, already bound, binds to the 5' end of the mRNA, and then moves along mRNA, until it reaches the start codon, thus form hydrogen bonds. The first components to associate with each other are mRNA, a tRNA bearing the MET, and the small ribosomal subunit. This followed by the attachment of a large ribosomal subunit, completing the translation initiation complex. There are two sites on the large ribosomal subunit, which are P sites and A sites. At the completion of the initiation process, the initiator tRNA sits in the P site of the ribosome, and the vacant A site is ready for the next amino acid tRNA. During elongation, amino acids are added, one by one, to the previous amino acids of growing chain. The ribosome read mRNA in one direction only, from 5' end to 3' end of mRNA. The step involve 3 process, codon recognition, peptide bond formation and translocation. During codon recognition, anticodon of the second amino acid tRNA pairs with the complementary second codon in A site. Then, a peptide bond between the two amino acids is formed. The reaction is catalyzed by our RNA molecule of large ribosomal subunit. Polypeptide from initiator tRNA in P site is removed and attached to the second tRNA in the A site. During translocation, ribosome moves along mRNA, translocating the second tRNA in the A site to P site. A site is occupied by the third amino acid tRNA. The final stage of translation is termination, when stop codon, which are UAG or UAA or UGA, in mRNA, reach the A site. A release factor, which is a protein shaped like an amino acid tRNA, binds directly to the stop codon in the A site. The release factor causes the addition of a water molecule instead of an amino acid to polypeptide chain. This reaction hydrolyzes the bond between the completed polypeptide and the tRNA in the P site, releasing the polypeptide through the exit tunnel of the ribosome's large subunit. Let's watch this video together.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something, so protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA.
You can access the questions by scanning QR code below. Thank you. And bye-bye.